Good morning everyone. Welcome back to my channel Pari's tutorial. I'm back with one more interesting video. Please let's get into the class. Hello everyone. Welcome back to my class. In the last class I have already discussed about the process of glycolysis that happens in the cytoplasm of all the living cells. Okay? And the conversion of one molecule of glucose into two molecules of pyruvate with a net gain of 2 ATP is also discussed in the class. In this class, I want to discuss about the regulation of the glycolysis. The regulation of glycolysis is mainly dependent on the presence of energy needed, requirement, and the importance of the enzymes, which really catalyze the irreversible reactions in glycolysis. Okay, so let's go for the class regarding the regulation of glycolysis. So glycolysis regulation, you know that glycolysis is a pathway which is an important pathway for the production of 2 ATP for the energy. But the complete conversion of glucose into energy happens after the Krebs cycle and finally the electron transport chain. I will let you know in the coming classes about those pathways in detail. Okay, so this glycolysis is meant for the generation of ATP and also it provides the building blocks for the synthetic reactions like fatty acids formation. Okay, and apart from that, you all know that there are three important steps in glycolysis where irreversible reactions happen. The irreversible reactions in glycolysis are mainly catalyzed by three important enzymes called hexokinase, phosphofructokinase, and pyruvate kinases. These are all the kinase family which are meant for the conversion or transfer of phosphate group from one molecule to another molecule. Right? If you remember, in the first step of glycolysis, there is a conversion of glucose into glucose 6 phosphate, which is catalyzed by a hexokinase enzyme. Okay, so all these important three irreversible steps are all regulating the glycolysis. The whole fate of glycolysis, or whether it has to go for end of the end of the production of pyruvate or not, is mainly dependent on these three important enzymes. So that is why they are called the regulatory enzymes of glycolysis. Okay. And before I enter into it, you have to be aware of a term called allosteric inhibition. What is allosteric inhibition? You all know that enzyme has got a catalytic site on it where the substrate would add and it will help you for the formation of the product. But what happens is, in an enzyme, which is a protein, has got an effector which would bind to the site that is opposite to the catalytic site. Like instead of binding to the original site, it would be binding to the opposite direction, which is called as an effector. And because of this binding, the whole conformation, all the protein nature, or the whole capability of the particular morphology of that particular enzyme is changed, and it's called as allosteric inhibition. The inhibitors get attached to the other side of your opposite side of your catalytic site instead of the pairing to the original side. So that is what the glycolysis is mainly regulated by the allosteric mechanism. Okay, based on which we can determine the energy requirements. Okay, so this enzyme catalyzes the first type of glycolysis, and it is inhibited by its own product. You know that glucose 6 phosphate is produced from glucose, because you are meant for trapping of that particular glucose molecule and undergoing a phosphorylation. So for that hexokinase would help. So what happens is, this is an irreversible step, right? So this glucose 6 phosphate is the activity of this particular hexokinase is inhibited by the glucose 6 phosphate itself. Okay, and a rise in glucose 6 phosphate, once if there is an increase in glucose 6 phosphate, obviously it would trigger the phosphofructokinase that communicates with the hexokinase. Yes, here phosphofructokinase is an important important enzyme in glycolysis, which would catalyze the conformation or the conversion of fructose 6 phosphate into fructose 1,6 bisphosphate. Right, it is also an irreversible. The rise in glucose 6 phosphate triggers the phosphofructokinase and communicates with the hexokinase, and due to which, when the phosphofructokinase is in inactive position, what happens? There is no conversion of fructose 6 to 1,6 happening. So fructose 6 phosphate would also be increasing the cell. And you know that fructose 6 phosphate and fructose conversion of glucose 6 phosphate to fructose 6 phosphate is catalyzed by an enzyme called isomerase. Being an isomer, as it is a reversible reaction, as fructose 6 is also increasing, glucose 6 phosphate would also increase, right, in equilibrium with the fructose 6 phosphate. That is how hexokinase and phosphofructokinase are togetherly regulating the cycle glycolysis. Hence, if you inhibit this particular phosphofructokinase, it will also inhibit the function of the hexokinase. Thus, they are both are interrelated. Okay, that is why the hexokinase is considered to be one of the regulatory enzymes of glycolysis. So, more importantly than hexokinase, we have phosphofructokinase, because phosphofructokinase is considered to be the pacemaker. So you have already heard that pacemaker is something that would regulate your heartbeat, right? So regulating your heartbeat is very important. In the same way, regulating your glycolysis is also important, which is done by phosphofructokinase. Okay, so what happens? You know that when glycolysis generally happens when you are short of energy, right? When you are not having any ATP in your cell, then the breakdown of glucose would happen, leading to the production of ATP, right? But in the same way, when ATP levels are more in number, like if you have much more ATP in the cell, the glycolysis need not happen. Like it's not an important thing to happen. That ATP can be useful for many other metabolic pathways that happen in the cell. So glycolysis need not happen because you have enough ATP. So for that, it is what really happens is when ATP in the cell increases, the glycolysis need to be shut down. For that, how this phosphofructokinase plays an important role, I am explaining you. And I told you the high levels of ATP allosterically inhibit the enzyme. Yeah, this is important. Allosteric activity is done by this enzyme in the step where fructose 6 phosphate is converted to fructose 1,6 phosphate with utilization of 1 ATP. Yes, so this is the committed step. The conversion of fructose 6 phosphate to fructose 1,6 phosphate is considered to be a committed step. It's an irreversible reaction that has to happen for the glycolysis to continue. But what happens is, because of ATP present in the cell, it will be immediately notified that presence of ATP would immediately have an allosteric effect on phosphofructokinase, making it, inhibit the process. So whenever ATP is lower, AMP would be higher. They are reversely proportional to inverse. So you know that ATP is inhibiting the process, right? Increase in ATP is stopping the process. AMP would reverse the inhibiting activity of your ATP. Okay, and I will tell you in the next slide itself how the mechanism pyruvate, citrate and ATP. Yes, this phosphofructokinase would allosterically, this is an enzyme, right? This phosphofructokinase is enzyme, okay, and to which allosterically what happens is it's inhibited by a presence of more amount of ATP, more amounts of citrate and more amount of phosphoenol pyruvate. When these three quantities are more in number, this PFK gets allosterically inhibited and leading to stop the glycolysis for the further happening. That is what you call feed forward stimulation. Okay, let's discuss now about what is feed forward stimulation. So this is in glycolysis is mainly done by fructose 2,6 bisphosphate. Okay, see, the enzyme phosphofructokinase help for the conversion of fructose 6 phosphate to fructose 1,6 bisphosphate. This is done by PFK1, but there is other class of enzyme called PFK2 which would catalyze the conformation or the conversion of the fructose 6 phosphate to fructose 2,6 bisphosphate, right? So you have fructose 2,6 bisphosphate and fructose 1,6 bisphosphate, but what happens is this fructose 2,6 bisphosphate would reduce the inhibitory effect. You know that ATP would reduce the effect of going for the glycolysis, but this fructose 2,6 phosphate would remove that particular inhibitory effect of the ATP, and thus what happens, this PFK1 has got a good affinity for the fructose 6 phosphate to convert into fructose 1,6 bisphosphate. Hence the glycolysis keeps on continuing. It's called as feed forward stimulation. Okay, so this is what I want to talk about. That is why PFK is very important for regulating. Okay, so what is next? Pyruvate kinase. So pyruvate kinase is the last enzyme that is involved for the ATP production, where the conversion of phosphoenol pyruvate to 2 molecules of pyruvate would happen, and there is an ATP generation also in the last stage, right? So that is called the kinase that would catalyze the final step, and it is mainly regulated by the allosteric effect covalent modifiers, and also some hormonal control. Okay, so allosteric effects, like I told you, pyruvate kinase has been found allosterically activated by fructose bisphosphate and allosterically inactivated by ATP and alanine. So this is alanine is a amino acid. Okay, this fructose bisphosphate binds to allosteric binding site. You have a domain of C where the fructose 6 phosphate would bind to it, and it would change the conformation of your enzyme. Thus, it would activate the PK activity. So, fructose bisphosphate also provides the feed forward stimulation, because the higher concentration of fructose bisphosphate increases the the allosteric modification of your pyruvate kinase. Hence, the keep converting into two molecules of pyruvate. So even covalent modifiers also regulates the cycle by undergoing phosphorylation, dephosphorylation, acetylation, and succinylation. Okay, these are the important regulators of your glycolysis. Okay, I told you AMP at high is also allosterically inhibited by citrate. Citrate is something that is a very first product of Krebs cycle, right? So if you have lots and lots of citrate in the cell, you don't need for the further glycolysis to happen, because you have already entered into the Krebs cycle and you have enough amount of acetyl CoA which has converted into citrate, and further enzyme conversion would happen, leading to the formation of ATP, FADH and NADH2, which would go for electron transport. That is, if you have amounts of citrate, further glycolysis is not needed. That is why this citrate would also, high amount of citrate would also inhibit the activity of phosphofructokinase, thus inhibiting the glycolysis. Okay, phosphoenol pyruvate, presence of citrate, presence of more amount of AT, all these things would allosterically inhibit the activity of phosphofructokinase, thus shutting down the process of glycolysis. That is how it is maintained in the cell. Okay, so hope you got an idea about how the whole glycolysis is maintained. Okay, now let's talk about the fate of pyruvate. So the pyruvate that we obtain in the cytoplasm has to now enter into the mitochondrial matrix for the further breakdown and production of carbon dioxide and all. So before that, let us know from where you are getting pyruvate other than glycolysis. Pyruvate we will be getting from glycolysis. Other than glycolysis, we have many other cycles happening in the cell which leads to the formation of pyruvate. So what are the sources of pyruvate? Let's discuss. So glucose is the main source of pyruvate, as you all know. Lactate in liver cells also convert into pyruvate through gluconeogenesis. I told you gluconeogenesis is the formation of the glucose from non-carbohydrate precursors, right? So in the same way, alanine is an amino acid. So by reaction called transamination, there is a production of pyruvate from alanine. In the same way, glycine, serine and cysteine, these are all the amino acids which undergo the transamination reaction, and the enzymes like serine dehydratase are all involved for the conversion into pyruvate. So these are all the sources of pyruvate. So now you have got source of pyruvate. Now you are meant for the conversion based on the presence of oxygen, availability of oxygen and all. Now that's how the fate of pyruvate is decided. Okay. Okay. I told you the presence or absence of oxygen would determine the fate of your pyruvate. Okay. So if more and more amount of oxygen is present, that is nothing but aerobic conditions, pyruvate is first converted into acetyl CoA, and later on it will be further reduced to carbon dioxide and water through Krebs cycle. They will enter the citric acid cycle, right? And if it is if the presence of oxygen is less, or you can say there is no oxygen available, they have to go for the fermentation. So I will tell you how it is. Here I wanted to show you that this is the glucose molecule. You got two molecules of pyruvate here. Okay, this pyruvate is source, and here from which we have oxygen. Okay, in the aerobic conditions, you will be releasing two carbon dioxide molecules with the production of two acetyl CoA, and this acetyl CoA would now enter into the citric acid cycle, producing the carbon dioxide and water. And this is the normal cycle that would normally happen in all the mammalian cells and all. This would generally happen in the yeast cells, all the fermentations and all. And this under anaerobic conditions, even in our cells like eukaryotic cells, or even in the mammalian cells, in our cells, this lactate food fermentation would happen, because whenever you go for a vigorous exercise, and all your glucose molecules are completely broken down into pyruvate and all, and pyruvate is finally converted into lactate under anaerobic conditions. When you are in exercising, obviously the oxygen would be in a lower concentration, like almost lower, or it would be in an anaerobic condition. During a anaerobic condition, you may also experience pains in your muscles and all, which is due to presence of the lactate fermentation. The lactic acid get accumulated in your muscle cells due to the over strenuous or exercises of vigorous contracting of your muscles would lead to the formation of lactate, because fermentation of lactic acid is happening in the muscle cells. So this is commonly happening in our cells. So based on the availability of oxygen, the fate of pyruvate is decided. Aerobic respiration. So aerobic respiration is, if oxygen is available, the pyruvate molecules produced at the end of glycolysis will get transported into mitochondria. In the mitochondrial matrix, pyruvate will be transformed into a 2-carbon compound, to which the coenzyme A is added. The coenzyme is generally made from the vitamin B5 that reacts with acetylic acid, forming acetyl-CoA. Okay, this resulting compound is called acetyl-CoA, and the acetyl-CoA is widely used for many variety ways in the cell, but it is mainly going for the citric acid cycle only. Okay, what is anaerobic respiration or the fermentation? So the fermentation generally happens in anaerobic, like in the area where oxygen is not available, right? So lactic acid fermentation, I told, is a type of anaerobic respiration that is carried out by yogurt bacteria, lactobacillus. Yes, this lactic acid formation, what it would meant for the conversion of it, a 3-carbon compound into a 3-carbon compound lactate, and regenerates NAD plus. Here this step is important, because in glycolysis you have already come across the conversion, or NAD plus gets reduced to NADH, right? There is a dehydrogenase that would act upon and help for the conversion of NAD plus to NADH. So for that, for NAD plus to be required in the cell, we get it from the anaerobic respiration. Anaerobic fermentation, we'll get lots and lots of NAD plus, which would be helpful for again the glycolysis to continue for the generation of energy. Okay, so NAD plus is present in the glycolysis, continue to make the ATP in lower oxygen conditions. So yeah, if there's a limited supply of NAD plus, I told you, is available in the cell, right? So here NAD plus acts as a reducing agent, generating NAD plus. Okay, so this is what I want to discuss about in this class. In the next class, I want to discuss about the important metabolic pathway called Krebs cycle or citric acid cycle, where the further breakdown of the glucose molecule for the energy production would happen. Okay, so by the end of Krebs cycle, almost the energy is completely obtained from glucose molecule. Maximum energy is being drawn from the glucose molecule, and finally they will enter into the electron transport, where the fate where there's a lots of production of ATP, final production of ATP. Okay, hope you understood the mechanism and regulatory enzymes in the cell. So see you in the next class, and thank you. I have also attached the main important here I want to discuss with you is I've also attached the important links regarding the structure of all the 3 enzymes involved in the glycolysis. Please go through it. And the pathway cycle and also the fate of pyruvate is everything discussed in the links. Please go through the slides and all. So hope it will be useful for you. Okay, thank you, and see you in the next class.